Laura is joining us from the Karolinska Institute, and she's going to be talking about sort of taking over from where we left off yesterday, gene and environment interplay in musicality and music acquisition. We focused really in on the phenomenon and overlap between reading and music yesterday, Laura, but very much talked about this nurture-nature interplay. So you sort of carry on where we left off yesterday, which is really lovely, also not by accident. So it's a nice continuation. Thank you so much for joining us. And you can start sharing your screen.
Perfect.
And it's over to you. I'll give you a 5-minute warning and in the chat, and there you go.
Perfect. Thank you very much for the introduction, and welcome everyone to this first talk today. My name is Laura Wessedlijk, and I have been doing research into gene-environmental interplay underlying musicality for the last 4 years, together with Frederick Ulen and Miriam Mosing at the Karolinska Institute, and nowadays at the Max Planck Institute for Empirical Aesthetics in Frankfurt. And today I will tell you about our research and about music and genetics. And I will start with talking about indications for why genes could be relevant for music. And then I'll tell you a bit more about how we can actually investigate whether genes are of importance with the use of twin studies. And next, I will speak about molecular genetic studies that can be used to identify the specific genetic variants that are associated with music or music-related behavior. And I will discuss several studies conducted by us, investigating gene-environmental interplay for musicality, and I'll conclude with a short summary. So the first evidence for a genetic basis for music skills comes from the fact that music is, since ancient time, present in all populations and cultures around the world. And if people from all around the world engage in music, it seems likely that musical engagement is a human trait that is part of our biology and not dependent on a particular environment. And another reason to suspect genetic influences on musicality comes from the observation that music skills aggregate within families. And in other words, if one family member is highly musical, it's more likely that other members of that family are also more musical. And one of the most famous examples of this is the Bach family, which consisted of more than 50 highly accomplished and well-known musicians. However, the fact that musical abilities cluster in families does not tell us exactly how important genetic influences are, because family members do not only share part of their genes, but also their common family environment. Nevertheless, it does suggest that there might be a heritable component to musical abilities. And lastly, we know about at least 2 genetically determined syndromes that cause both cognitive impairments, but also alter individuals' music skills. The Williams-Beuren syndrome, which is caused by a heterozygous gene deletion on chromosome 7, causes serious cognitive deficits, but with contrasting good music skills and a strong interest in music. And on the other hand, another syndrome caused by a rare mutation also on chromosome 7 results in severe language problems and reduced music skills. So altogether, these observations seem to suggest that musicality and musical engagement are at least to some degree influenced by genetic factors. And we can determine the exact degree to which genetic factors are of influence by making use of twin data and genotype data. And twin studies have been around the longest, since the early 1900s. And they utilize the existence of 2 sorts of twins. There are monozygotic twins that develop from one zygote, sharing approximately 100% of their DNA, and dizygotic twins that develop both from their own egg that got fertilized by their own sperm cell. So they, as regular siblings, share on average 50% of their DNA. But both types of twins, if not reared apart, share their common environment, both growing up in the same family environment. And this allows us to make some inferences, because if the resemblance between monozygotic twin pairs is higher than within dizygotic twin pairs, we infer that genetic factors must play a role. And if the correlation between monozygotic twins is not double that of dizygotic twins, the shared family environment must play a role, because the twin pairs are then more similar than would be expected based only on genetic influences. And everything in which monozygotic twin pairs differ is due to unique environmental influences. And we use structural equation modeling actually nowadays to estimate the degree to which genetic, family environmental, and unique environmental influences can be calculated on any behavior of interest. And these models can be expanded. I'm showing here the most basic classical twin model, but they can be expanded taking gender into account, including moderators, and I'll come back to this later in the talk. But I will now tell you about the twin studies that have been conducted on musical phenotypes. So this is an overview of the twin studies that have been conducted on music skills or music achievement, music-related behavior. And overall, even though the exact amount of genetic influences differs on the aspect of musicality that is measured, the age range of the twin sample and the gender composition, because for some musical phenotypes, the heritability estimates were different for males and for females, and sometimes not. Twin studies consistently show that a broad range of music skills and music-related behavior have a considerable genetic basis. It ranges from 13 to 86%, with an average around 40 to 50%. Genetic influences seem to be more important for music skills and aptitude than for lifetime musical achievement or engagement. And additionally, we see that for men, genetic factors are of more importance than for women for the majority of the studies. So after decades of twin studies showing that almost all human behavior and also music-related behavior is to some degree influenced by genetic factors, molecular genetic research evolved after the discovery of the DNA helix in 1953. So our genetic makeup is coded in DNA, and the human DNA contains 4 to 5 million single nucleotide polymorphisms, SNPs, or genetic variants. And in the 1980s, family linkage studies were the first to link genetic regions to human behavior at the molecular level. However, this approach turned out to be successful only in exceptional cases, namely for traits that are influenced by genes with very large effect sizes, such as single genes disorders. And in the early 2000s, the first whole genome was sequenced. And at first, researchers tried to link behavior to genetic variants chosen based on a priori hypothesis in candidate gene studies. However, it soon became apparent that there was not enough understanding of the genetic architecture of human diseases and behavior to come up with good a priori hypotheses. Because more importantly, human behavior is polygenic, meaning that a trait is influenced by more than one gene. And additionally, genes are pleiotropic, which means that one gene influences more than one trait. So genes are large in quantity and small in effect, and therefore candidate gene studies are extremely prone to false positive findings and have encountered replication problems. But over time, insight into the underlying human genetic architecture was gained, which allowed for measuring more genetic variants in individuals for less money. And this resulted in the first genome-wide association study, or GWAS, in 2005. And in a GWAS, each measured and imputed SNP, or genetic variant, is tested for its association with behavior of interest. So one group with depression versus a group without depression, for example. So this leads to an enormous amount of tests. So rigid adjustments for multiple testing need to be performed. But nevertheless, this is a hypothesis-free design, which has resulted in many new and replicable associations between genetic variants and human behavior or diseases. And since the first GWAS in 2005, post-GWAS approaches developed to further study the genetic variants that were discovered by these GWASs. For example, the Polygenic Score approach, which uses the estimate estimated effect sizes of the SNPs as identified in the GWAS, and it uses it as weights to calculate for every person a personal score representing his or her genetic propensity for the behavior studied in the GWAS. So this molecular genetic field, what has been done for music skills? And although the field is still in its infancy, the first studies aiming to reveal specific genetics relevant for music skills also emerged. And as in most research fields, the earlier studies were candidate gene and linkage studies, which we know nowadays to suffer from replication problems and are hampered by small effect sizes. However, recently also the first GWAS was performed on a musically related phenotype. So in this GWAS, the authors compared the genome of individuals who reported they can clap in time with a musical beat to those who cannot. And although this single question is obviously limited as a measure of music skills or musical aptitude, it does allow us to take a look at the genetic architecture underlying a music-related phenotype. And this GWAS identified 69 genetic loci that influence variation in self-reported musical beat synchronization ability, which means that this trait, like many others, is highly polygenic. Many genetic loci are involved, and it is also pleiotropic because many of these genetic loci are also involved in other human traits. And interestingly, none of the past findings from candidate gene studies or linkage studies were actually found and replicated in this GWAS. So that there is a genetic basic for music skills is well established by now by both twin studies and molecular genetic studies. And it will only be a matter of time until more large GWAS studies and post-GWAS approaches reveal the genetic variants involved in different aspects of musicality. However, as for almost all human behavior, both genes and the environment work together in the development of music skills. And it is therefore important when studying the etiology of musicality and its relation to other traits to take into account both genetic and environmental influences. And other than providing heritability estimates or identifying relevant genetic loci, twin and genotype data can also provide a useful tool to study environmental influences free from genetic and familial confounding, as well as gene-environmental interplay. So we speak of gene-environment interaction when the effect of the environment depends on an individual's genotype, or when the effect of a gene depends on an environmental exposure. And we speak of gene-environmental correlation in case there is a relation or a correlation between genetic and environmental influences. And there are three different types of GE correlation. There's passive gene-environment correlation, which refers to the association between the genotype a child inherits from its parents and the environment in which the child is raised. So, for example, parents pass on their genetic predisposition for musicality and provide their offspring with a musically enriched childhood environment because of their own music interest. So reactive gene-environmental correlation is when an environmental condition provided to an individual is a reaction to the individual's genotype. And active gene-environment correlation is when exposure to environmental conditions depends on an individual's genotype. So, for example, parents might notice their child's interest in music, partly because of genetic reasons, and they react by providing the child with music classes, or the child actively requests it. So both twin and genotype data can shed light on these phenomena, either by extending the classical twin design, which I showed you in the beginning, by including moderators, for example, or multivariate approaches, or, for example, by constructing polygenic scores and testing for interactions or correlations with environmental factors. So our lab makes use of data that were initiated by Fredrik Ullén at the Swedish Twin Register in 2011 for the studies Humans Making Music, to investigate genetic influences and gene-environmental interplay for musicality. So during this data collection, twins were tested on their musical abilities with a melody, pitch, and rhythm discrimination test, which they did online. But they also answered many questions about music practice, music listening, how musically enriched their childhood environment was, but also other things like IQ, depressive symptoms, personality traits. And we use these data to investigate relationships between variables of interest in music skills or musical achievement, and also to investigate gene-environment interplay. And so, for example, one of the first studies that looked using this sample looked at the relationship between practice and music skills in adulthood. And besides thoroughly investigating the causal relationship between practice and music skills, controlling for genetics and shared familial influences, this study showed that not only music skills themselves are influenced by genetic factors, but maybe more surprisingly, music practice, which we see as an environmental factor that influences people's music skills, in itself is also highly influenced by genetic factors, actually to a similar amount as music skills themselves. So this suggests that this environmental factor is not so purely environmental after all. And it is a clear example of gene-environment correlation, because the amount someone practices is also influenced by his or her genetic predisposition. And another different example comes from one of our studies investigating the relationship between growing up in a family which is musically stimulating, a musically enriched childhood environment. So I tested that or estimated that depending on the number of records available in a family home, how many individuals in the twins family played music, how often they visited concerts, and whether they were offered musical education before the age of 12. So this measurement of how musically enriched the childhood environment was, what is the effect of that on music skills in adulthood and musical achievement in adulthood? So I extended the classical twin model with a moderator, namely the level of musical enrichment. And so I found that there is an association between the childhood environment and the level of musical achievement in adulthood. So there's this main effect. Clearly, the more enriched the childhood environment, the higher level of musical achievement in later life. But we did not only see this main effect, we also looked at the estimates of genetic and environmental influences on musical achievement. And we saw that they also changed depending on the childhood environment. So here you see in this figure that the higher the musically enrichment of the childhood environment, the larger the total variance. So the more individuals differ from each other. And this was largely driven by A. So A is the genetic factors. So genetic factors mostly drive this amplified differences. So in other words, a musically enriched family environment amplifies individual differences and can offer the opportunity for individuals with a genetic predisposition for musicality to achieve higher levels of expertise. So we see that the heritability of musical achievement varies by the environment. And this is an example of gene-environment interaction. So lastly, I want to give you one more example of gene-environment interplay that comes from one of our recent studies using molecular genetics instead of the twin data. So we used this GWAS, which I talked about earlier, and we created polygenic scores for the big twin sample that we have. Because in this sample, we have a lot of information available about individuals' music skills, achievement, their music-related behavior. So this polygenic score represents an individual's genetic propensity or vulnerability for self-perceived musical beat synchronization. And it's calculated as follows. As you can see in the bottom of this picture, the effect sizes of each SNP or genetic variant calculated by the GWAS is weighted depending on the SNPs or the DNA of the participants. Then it's added up together, resulting in one overall score per individual, which represents the genetic propensity or vulnerability to the trait which was measured in the GWAS. And that, in our case, is a music-related trait, beat synchronization. So every person has an individual score on a distribution where they are regarding their genetic vulnerability. So we tested whether this derived polygenic score explains variance in musical traits of interest in our target sample. And of course, we were most interested to look at rhythm ability. And we saw that the polygenic score, so the genes underlying self-reported beat synchronization, indeed significantly predict individuals' objectively measured rhythm discrimination skills. So they were tested with an online test on their rhythm skills. And this is predicted by this polygenic score. But this polygenic score actually also predicts other music skills like melody discrimination, pitch discrimination, overall aptitude, but also interestingly, variables music related, like listening to music, the age you started playing music, flow proneness in the music domain. And in contrast, the polygenic score was not significantly associated with any of the non-music related control variables, such as sport engagement or achievements in other domains. So therefore, we concluded that this polygenic score seems to tap into the genetic architecture underlying the broader music domain with quite good discriminant validity. And what we can actually learn from this regarding GWAS approaches is that despite from it being far from an ideal phenotype, it seems to tap quite well into the genetics of the broader music domain and music abilities. So it highlights the importance of power and large samples for gene finding, and possibly more important than a really good and precise phenotype. But what is of interest is that we also use this polygenic score to look if it correlates with this musically enrichment of the childhood environment, which I talked about before. So we indeed found also, again, a small but significant correlation between people's polygenic score and the musical enrichment of the childhood environment. So in other words, individuals with a genetic predisposition for music also more often grow up in a musically enriched childhood environment. And this is another example of gene-environment correlation. The environment someone grows up into is related to his or her genotype. And whether this is due to passive, evocative, or active gene-environment correlation, we cannot disentangle. But overall, this study also shows, using molecular genotype data, that there's the presence of gene-environmental interplay in musicality. So this brings me to the end of the talk, and to summarize, we know by now that musicality and music-related behavior has a considerable genetic basis, and that both genes and the environment are of importance. And molecular genetic research only just started to reveal genetic variants relevant for music skills, and many of the earlier gene finding studies were limited by small samples and replication problems. However, the first well-powered GWAS identified 69 loci associated with self-reported beat synchronization ability. So, given the complexity of musicality and its many facets, more large GWASs are needed to provide a better understanding of the genetic variants relevant for music. But I hope that I somewhat convinced you that for a complex trait such as music, and like for many other complex traits and behaviors, the nature versus nurture dichotomy is false, and genes and the environment work together, not necessarily independently. And twin and genotype data can be used for more than just estimating heritability or identifying genetic loci. We can use them and should use them to study gene-environmental interplay and to study environmental influences on musicality and music acquisition while controlling for genetic confounding, which is necessary. So overall, the genetic research to date highlights the importance for music research to consider both the environment and familial, so the family environment and genetic factors, as well as the correlations and interactions between them. So I would like to really finish by acknowledging my colleagues and supervisors, Frederick Ulen and Miriam Mosing, and of course, many more wonderful colleagues and collaborations and access to great data that make this research possible. Thank you for your attention, and I'm happy to answer any questions.
Thank you so much, Laura, for a wonderful talk. I adore your logo more than I can say. I've been staring at it in the corner, but
now it's big out. Yeah.
It's fantastic. And thank you so much for the great talk, which, as well encapsulated by your logo, I think gives us a really good sense of why we are having this conference, to really try and understand what makes us like and enjoy and indeed be good at music. So I have a couple of questions myself. I'm waiting for the YouTube live stream to catch up with the end of the talk. So as we wait for people out there to provide us with their thoughts and feedback, I have a couple of questions for you, probably more than I should. You didn't mention the words experience or expertise, but I think that they're sort of captured by some of the other concepts that you mentioned. So I wanted to know whether you could speak to this idea, and maybe with respect to some of the loci you mentioned, is there a sense of whether these genes are coding more for, say, sensory motor coordination, or are they more on the perceptual side? Are they therefore going to be better at helping us become experienced musicians, or help us to practice, which is what I think the experience side is more about? Or are they going to be better at the expertise side of things and make us just better at perceiving, being more likely to be absolute pitch? Or so do you have a sense of that dichotomy, as it were?
Yes, thank you. That's a very good question, and I'm sorry, but I have to answer it with that we do not know yet, because the phenotype, the self-reported can you clap to a beat, is, though it captures really music-related behavior, it's of course not a perfect measurement of expertise versus practice versus experience. So I can't yet
more to come.
Yes, it's really more GWASs are needed to really identify the specific genes related to the specific aspects of musicality. This is more a very broad overarching.
That's fine. This is why we're here. There are questions coming in. As I told you, that they're just a little bit behind. So RA says, I'm curious, did the polygenic score of the last study that you presented correlate with working memory or executive control or some language related ability? And this is from Ria Sano.
Yes, that is a very interesting question, which we would love to look at. I have looked with IQ. But that is the only, we didn't have, because it's a sample which is really deeply phenotyped on music behavior, but not on language. And there is a follow-up data collection happening this year with a lot of extra questions about language behavior and language capacities, cognitive abilities. And then we'll be able to look at that. But for this study, we haven't been able to test that.
That's yeah. I think this question also relates nicely to, as I said, we had a talk by Shruti Nayak yesterday, who was talking about that shared music ability and reading. So we have another question from Music Cognition. What are the realistic next steps in this research, or in other words, the low-hanging fruit?
Well, I think more GWASs will be the next realistic step, but also post-GWAS approaches. So better analysis to really zoom into the genetic variants to actually be able to more specifically link them to specific music-related behavior. I think that's what the next steps will be.
Fantastic. And we've perfectly timed this so that we've come to the end of your allotted time, and we have Pat Savage in the background looking like he's about to go and have a really good time. Instead, he's just going to have a pretty good time with us. So it just leaves me to thank you again, Laura, and obviously Miriam, for your fantastic talk and for giving us a really good backdrop to these GWAS questions. Thank you so much for that great introduction. And thank you for your time. If anybody has more questions for Laura or any of our other speakers, do turn to the YouTube chat. You have a lot of applause from some of your collaborators and others. So go and enjoy your praise. And